In this lecture, I'd like to introduce population analysis using statistical genetic tool. So why population analysis is important? That's because characterizing population has a useful implication in a wide range of fields. For example, medicine, human health, biology, and other many sciences. And it is essential tool for research as well as practical application. So this should be useful in your research or when you have a professional job. In order to characterize population, population parameters are key components. So one of the key components is genetic parameters, for example, heritability and genetic correlation. So let's have a look what is population. It is any large collection of individuals, for example, Australians. And what is parameter? That is any summary number or statistic characterizing the population. For example, it can be mean or variation. So this variation is important to characterize population. Then I will mostly focus on how to estimate variation, especially for genetic variation or genetic variance. And one of important the term is sample. It is a representative group randomly drawn from the population. For example, set of 1,000 individuals from Australian. Variation or variance is one of the important parameters to characterize population. It is fundamental in evolutionary biology. It is also important implication in wide range of practical fields like medicine, population health, selection program in livestock and plants, and so on. So I will give a simple example about variance. So I will use some whiteboard. So if there is, for example, thousands of sample from the populations, so let's assume we measure their height. So also we assume there are 1,000 men. So we can range according to their height. So, so this will be 150 centimeters to the 200 centimeters. Then the middle is 157, for example. Then when you make a histogram, then this middle, the value should have highest density as expected. Then slightly lower value, for example 160 centimeters, then you have probably less density, less number of individual for this. Also a bit higher, 180 or 190, then also you have some numbers probably smaller than middle, but higher than the extreme two tails. So for the extreme two tails, probably you found a very small number compared to the middle range, right? So when you get the histogram, then you'll get the normal distribution. So, so this point is mean. This is the mean, often called the mu. Then the range between some point. So let's have a line here. Then this range, that's around 68%. This is according to statistical theory, 68%. Then, sorry. So this is one standard deviation. And standard deviation is the square root of variance. In other words, variance equals standard deviation squared, right? Then two standard deviation, the range of two standard deviation is around 95% of the whole population. And now this is two standard deviation. And now three standard deviation is almost all population. This is 99% of the population. So that now makes sense what is the variance according to their distribution. So this is a simple example of a human height, but it can be applied to most of complex traits. In population analysis, key approach is to estimate variance. So analysis of variance, called ANOVA, is widely used in many fields. So this is developed by Fisher. So he is known as the founder of the modern statistics. So in order to estimate variance, there are a few terms you may need to know. Phenotypes, noted as P, that's individual observable characteristic or traits, for example, individual height. And genotypes, as G, the genetic makeup of an individual that determines phenotypes. So not only genetic effects, also there is residuals, that is unexplained errors. So phenotypes is consisting of genetic effects and unexplained errors. Also, phenotypic variance is decomposed as genetic variance and residual variance. So heritability, this is one of most important population parameters or genetic parameters, is genetic variance scaled by phenotypic variance, that is the sum of genetic variance and residual variance. So variance is often noted by sigma squared. So this is a genetic variance and this is a phenotypic variance. So in this picture, the individuals are from the same family. But for example, for coat color, like a trait, one is brown, but another is black and also brown. So there is a variations. So it is interesting to find out what it caused this variation, how much proportion is explained by genetics, how much is explained by the residual variance. Therefore, it can determine heritability, or heritability can be estimated from the some statistical tool. So in order to characterize population, one of the essential approaches to estimate heritability. Also, there are a number of other important genetic parameters, for example, genetic correlations. But I will first focus on heritability, how to estimate, then what should be needed to estimate heritability. So the first question is how to estimate. So I will explain briefly with some whiteboard. I will bring here. First, I delete this one. Yep. So as shown in the previous slide, so phenotypes is function of decomposed as G with the residuals, and we assume there are samples from population, that is individual 1 to n. Then each individual has measured phenotypes, right? So we call Y one for the first individuals and Y for n for the last individual. Then also genetic effects from first individuals and genetic effects from last individuals. Also there is residual effects last individual. So if we know each value for each sample for phenotypes and genetic effects and residuals, then it's straightforward to get the heritability. That's just a simple function of the variance of this vector of G, that we call genetic variance, divided by the variance of G plus variance of E, according to the equation in the previous slide. That's simple. But in real situation, the problem is you never observe this G through G. Also, you don't know this vector of residual values. What you can observe is only these phenotypes. So, given these phenotypes that are observable, you should disentangle between these genetics and residuals and estimate genetic variance and residual variance. Therefore, you can estimate heritability. So in order to this, given the phenotypes, you need some designs. Right, so there are a number of useful designs. That is MZ twin design, especially in human study. This is widely used. And DZ twin or full SIP design. So DZ and full SIP, the kinship coefficient is same. That's half of the gene between pairs sharing from their parents. And in the experimental species or livestock, also half-SIP designs is widely used. And granddaughter design is a special case of a half-SIP design. And there are also other designs based on complex pedigree. And designs are not always balanced. There can be unbalanced designs. For example, within each family, the number of members can be different. So this is a simple example. So for example, if you have the number of MZ twin pairs, then you have their phenotypes, for example, their height or their BMI or their weight. Then you can observe their phenotypes. Therefore, you can get the phenotypic correlation. There is a correlation between this first MZ twins and this second MZ twins. That's simple, right? Then this phenotypic correlation can be scaled by sample correlation. In this case, sample correlation is 1. That's because the MZ twins, they have exactly same genetic property. Their genotypes are same unless there are mutation. So in this design, heritability is the same as phenotypic correlation because sample correlation is 1. How about when using DZ twin or full SIP? So in this case, the sample correlation is a half, 0.5. That is the same as the proportion of gene they share between the SIPs. So as same as MZ twin design, also you can get phenotypic correlations. Then this can be divided by 0.5. That is the sample correlation between full SIP or DZ twins. Therefore, heritability can be estimated. And this is a half-SIP, actually granddaughter design. So there are a number of sires, and there are a number of dams per each sire, and there are a number of offspring, in this case daughters, per each dam. So in experimental species or livestock, this half-SIP design or granddaughter design is widely used and estimate heritability from this design. In practical, you will estimate heritability using twin design, full SIP design, or half SIP design based on R code that can be downloaded from the web. The information is in the practical material, including videos. Also, there are practical instruction videos. So here is the link. So each is around 10 minutes long. Then hopefully it can help you understand how to estimate heritability based on such designs. And also there are a number of R code that should be useful for your practical, as well as also it can be actually applied to your research when you're doing your own research. And it would be great if you give any feedback that will improve the lecture and practical materials. Thank you.